Okay, now let us proceed with the next topic, that is RNA processing. Of course, we already discussed the details of different types of RNA processing methods, including the different splicing mechanisms. But now right now the focus is especially on the regulatory mechanisms or gene regulation mechanisms, which is based on RNA processing. And you know that alternative RNA splicing can generate different mRNA molecules from the same primary transcript, which means organisms can produce more than one polypeptide from a single gene. And we already discussed the example of different alternative splicing mechanisms, including the immunoglobulin genes, in which immunoglobulin diversity is generated by alternative splicing and gene shuffling and other things. Right now, we have to understand that the RNA processing enables different products or polypeptide gene products to be produced from the primary transcript using these alternative splicing mechanisms. So this is one form of gene regulation in case of eukaryotes. Then we come to the post-transcriptional control of gene expression. In case of post-transcriptional control, as the name indicates, it is a post-transcriptional regulation after the transcription. So after the transcription, as you know, there is a primary transcript which needs to be processed appropriately to produce the processed mRNA, which is incorporated with the 5' cap, poly-A tail, and all other things. And the main focus of this post-transcriptional gene control is on two items, that is mRNA stability and mRNA degradation. The mRNA degradation, maybe we will discuss various items like poly-A tail and miRNA, RNAi or RNA interference, which is also known as gene silencing by RNA interference, and which will be coming under the mRNA degradation topic. So when we come to the mRNA stability, the classical example is casein mRNA stability, the case of casein mRNA stability. Here you can see that mammary gland tissues are stimulated by prolactin, and the synthesis of a casein protein increases dramatically in that case. If the mammary gland tissues were stimulated by prolactin, it increases the casein protein production dramatically. And it is not due to the increase in the rate of transcription, but it is due to the increase in the half-life of the casein mRNA. So this is one example of gene regulation by mRNA stability, or by influencing the mRNA stability. So in the case of casein, the example of casein, the addition of prolactin to the mammary gland tissue increases the mRNA stability, or the half-life of the mRNA is produced to produce or for the transcription of casein. And subsequently, that increase in the half-life or the stability of the mRNA of the casein increases the gene expression. And another example is a transferrin receptor mRNA stability. In mammalian cells, control ion concentration, or mammalian cells actually, they control the ion concentration by regulating the amounts of two proteins, an ion import protein called transferrin receptor and an ion storage protein called ferritin. The 3' untranslated regions of the TfR mRNA or transferrin mRNA contains 5 stem loops called ion response elements, or IREs. Deletion of two or more of these IREs in tandem or in random causes the loss of response to ion. So the transferrin, actually it is intended, or transferrin receptor is intended to import ion, or it's an ion import protein. And some of the IRE elements, or out of the 5 IRE elements present in that gene, TFR gene, if 1 or 2 were deleted, these IRE elements were deleted, then that results in the loss of ion uptake or ion import. When the ion concentration is high, the TFR mRNA decays rapidly. When the ion concentration is low, the TFR mRNA, it decays much more slowly. So the ion concentration influences the uptake of ion, or at a high concentration of the ion, TFR mRNA rapidly decays, and at low concentration, its decay is, or its half-life is much more. So its decay is low, that means its half-life is more. So that the mRNA will survive for long, and that results in the increased uptake of the ion. So the ion concentration influences the half-life of the mRNA or the stability of the mRNA, and in cases of low concentration of the ion, then mRNA stability is more, which results in increased uptake of the ion, so that it will be compensated. And if the ion concentration is high, what happens is that mRNA stability is low, so mRNA will be rapidly degraded and further uptake of the ion will not be there. So there is a compensating mechanism for ion uptake. So this difference in mRNA stability is about 20-fold, and it is much more important in determining the rate of accumulation of the TFR mRNA than the difference in the mRNA synthesis. That means the mRNA stability contributes about 10 times more than that of the mRNA synthesis. So that is better regulated with the mRNA stability regulation, not with the new mRNA production. So the regulation based on new mRNA production is only about, which is controlled only 2-fold, whereas the mRNA stability based regulation is about 20-fold. So that is a better regulatory mechanism for controlling the ion uptake. Okay. Now when we come to the mRNA degradation, eukaryotic mRNAs can have a survival time measured in weeks compared to the 1 or 2 minutes in case of prokaryotes. Okay, then how it is degraded? One is shortening of the poly-A tail, and another one is a removal of the 5' cap, and that allows nucleases to degrade the mRNA. As we discussed earlier, the poly-A tail and 5' cap gives protection to the eukaryotic mRNAs. So the shortening of the poly-A tail and removal of the 5' cap allows the nucleases to attack the end portions of the mRNA, and that becomes results in the degradation of the mRNA. The microRNAs can degrade mRNA or block its translation, and it is called RNA interference. So another mode of degradation of the mRNA, eukaryotic mRNA, is interaction with the microRNAs or miRNAs. So that can degrade the mRNA and block its translation. So the mRNA degradation can be shown using this diagram. You can see that in the first stage, the microRNA or miRNA precursor folds back on itself and held together by hydrogen bonds. You can see that this is a self-complementary region of the miRNA, and that self-complementary region will pair together and leaving a loop here, and this is the miRNA that is pairing on, or it folds on itself, and this pairing is formed. Now, an enzyme called dicer moves through the double-strand RNA cutting it into shorter segments. So a dicer molecule or an enzyme, it moves through this RNA and it is cut into smaller fragments. One strand of each of these short double-stranded RNA is degraded, and the other strand, which is known as mRNA, then associates with a complex of proteins. So from these fragments, one strand is degraded and the other one, out of this one strand is degraded and the other one will pair with a complex of protein, and this is the protein complex with the mRNA. And this complex, it bounds with this mRNA, can bound or it can base pair with any target mRNA that contains a complementary sequence. So it can bind to any mRNA which is having this complementary sequence. And the mRNA protein complex prevents gene expression either by degrading the target DNA or blocking its transcription. So there are two possibilities. Either it degrades the target mRNA or it blocks the translation by the ribosomes in the case of this mRNA. So it can block translation or it can degrade the target mRNA using mRNA or miRNA interference. So this is how mRNA is degraded, and it regulates the expression of a target RNA using these miRNAs. So mRNA can be degraded using miRNA interference. And this is another gene silencing by RNA interference. You can see that here we have a precursor molecule, RNA molecule, and a dicer enzyme will cleave this precursor molecule, and this is the sTRNA or small temporal RNA, and that is cleaved, and one strand of the sTRNA is now generated, and this is the mRNA to be silenced, and at complementary regions of the target mRNA, this sTRNA will bind, and that can lead to the degradation of the RNA or translation inhibition, as we described in the previous case. So here is another example in which we have a duplex RNA, and again the dicer enzyme will cleave it into siRNAs. siRNAs were also known as small interfering RNAs. So this siRNAs is again one strand of the siRNA is removed or it is degraded, and the remaining strand will pair with complementary regions on the target mRNA, and the result is the degradation, either degradation of the target mRNA or inhibition of translation by RNA interference. So the RNA interference can be based on small temporal RNAs, which is known as sTRNAs, or small interfering RNAs like siRNAs, or miRNAs, which is actually described as in the previous case, that is a microRNAs. All these work in the similar fashion. That means a dicer molecule or dicer enzyme will cleave that RNA molecule into smaller fragments, and after that one strand of that RNA is removed, and the remaining portion, which will be miRNA, sTRNA, or siRNA, that can interact with the complementary regions of the target sequence in the mRNA target. Then that mRNA target, when it interacts with the miRNA or sTRNA or siRNA, that will be subjected to degradation and translation inhibition of that concerned mRNA, and that results in the effective regulation of that particular gene expression. Okay. So in case of mRNA translation, the initiation of the translation can be blocked by regulatory proteins that bind to the untranslated regions and block the attachment of the ribosomes to the mRNA. So at the regulation of gene expression at a translation level or mRNA translation level, you can see that initiation of translation can be blocked by the regulatory proteins that binds to the untranslated regions. So in case of mRNAs, we have 5' and 3' UTRs or untranslated regions at both ends. So a protein complex or an inhibitor or a regulatory protein that binds to these initial untranslated regions will block the binding or attachment of the ribosomes and subsequent translation of that mRNA. So the 5' UTRs especially that will be attached with some regulatory proteins or some inhibitors, and that inhibition will block or that will prevent the ribosome assembly formation with the mRNA, and that results in the regulation or blockage of mRNA translation. And eukaryotes have at least 3 main mechanisms of translational regulation. One is phosphorylation of initiation factors by a number of protein kinases reduce activity and cause a general depression of translation. So the first one, phosphorylation of initiation factors by a number of protein kinases, then that reduces the activity or translation activity and cause a general depression in translation. Phosphorylation of initiation factors. The second one, translational repressors are proteins that bind directly to the mRNA, specifically or specific sites in 3' UTRs or untranslated regions, and they interact with the other translation initiation factors or 30 or 40S ribosomal subunit to prevent translation initiation. So the second type is translation repressors or repressor proteins, and these proteins bind directly to mRNA untranslated regions, especially the 3' untranslated regions, and also they interact with the other initiation factors or 40S ribosomal subunit and prevents the translation initiation. Okay, the third category is binding proteins, and in eukaryotes from yeast to mammals you can see, and these binding proteins disrupt the interaction between elongation factor eukaryotic initiation factor 4E and eukaryotic initiation factor 4G. So binding proteins, that is the third category, and they disrupt the interaction between initiation factor 4E and 4G in eukaryotes. The mammalian versions are known as 4E BPs, or you can see that eukaryotic initiation factor 4E binding proteins, that is a 4E BPs. And they limit the translation by binding to the site on eukaryotic initiation factor 4E when cell growth is low. So these 4E BPs, they limit the translation by binding to the site on eukaryotic initiation factor 4E when the cell growth is slow. Then when cell growth resumes or increases in response to growth factors or other stimuli, the binding proteins are inactivated by protein kinase dependent phosphorylation. So this is another representation in which the translational regulation of eukaryotic mRNA is shown. Here, one of the most important mechanisms for a translational regulation in eukaryotes that involves the binding of translational repressors, that is RNA binding proteins, to specific sites in the 3' untranslated regions. We already discussed it in the previous slide. So translation regulation by binding of some specific proteins to the 3' untranslated regions or UTRs of the mRNA. Here you can see this is the translational repressor molecules which binds to the 3' end. This is the 5' cap. This is the 3' end of the mRNA, and this is the 3' untranslated region which is shown in green, and this is the 5' untranslated region, and there are some translational repressor molecules which binds to the 3' untranslated region, and then effectively it interacts with this with the ribosome assembly and these proteins interact with the eukaryotic initiation factors or with the ribosome to prevent or slow the translation. So these translational repressors which binds here will interact with the initiation factor or the ribosome assembly to slow down or halt the translation process. So this is how the translation regulation is happening in case of a eukaryotic mRNAs. So generally, once again, I am explaining here. This is a 5' cap. This is a 5' untranslated region, and this is the translated region. Starting from the AUG codon, this region will be coded, and this is the 3' or 3' untranslated region or UTRs. And here you can, as you can see, the translational repressor molecules will bind to the 3' or 3' untranslated regions, and after binding, it will interact with the eukaryotic elongation factors 4E and 4G, and also with the ribosome assembly, and that interaction will result in the prevention or slowing down of a translation. So this is an example of a translation regulation in case of eukaryotic mRNAs. Eukaryotic mRNA lifetimes are relatively long compared to the prokaryotes. Means it may be hours to weeks. So there is more opportunity for translation control than in prokaryotes. The alpha subunit of the eukaryotic initiation factor 2 is a favorite target for the translational control. In heme starvation reticulocytes, heme controlled repressor or HCR is activated, so it can phosphorylate eukaryotic initiation factor 2 alpha and inhibit initiation. In virus infected cells, another kinase, that is DAI, is activated, and it also phosphorylates eukaryotic initiation factor 2 alpha and it inhibits translation. So there are some cases where such factors, such kinases or some other factors which interacts with these eukaryotic initiation factor alpha, and that results in the inhibition of translation initiation. Then ferritin mRNA translation is subject to induction by ion. This induction seems to work as follows. A repressor protein, aconitase apoprotein, that binds to a stem loop ion response elements or IRE near the 5' end of the 5' UTR of the ferritin mRNA. Ion removes this repressor and allows the translation of the mRNA to proceed with. Then another one is polypeptide processing. Any interference in the processing of a polypeptide can alter the gene expression. Polypeptides are processed through cleavage, chemical modifications, and protein transport to its target destination. So, regulation at the stage of polypeptide processing can be of different categories, that is by cleavage, by chemical modifications, and even at the stages of protein transport to its target destination. Degradation of the proteins, that means the lifespan of a protein varies and is strictly regulated by other proteins. Protein tagged with the ubiquitin are recognized by proteosomes and they are degraded. Then protein degradation, which is actually the final stage of the gene regulation, it is described here. The multiple ubiquitin molecules are attached to a protein by enzymes in the cytosol. So this is a protein to be degraded. Ubiquitin molecules will bind to that protein to be degraded. Then ubiquitin tagged protein is recognized by a proteosome which unfolds the protein and sequesters it within a central cavity. So a ubiquitin tagged protein, actually it is recognized by a proteosome or an enzyme complex, and it is actually trapped within the central cavity of that proteosome. The enzymatic components of the proteosome cut the protein into smaller peptides and which can be further degraded by other enzymes into the cytosol. So finally it is degraded into smaller fragments, and the proteasome and ubiquitin to be recycled for further use. So this is the degradation of or degradation mechanism, or ubiquitin mediated degradation mechanism of proteins. Okay, now we can conclude this session on gene regulation, starting from the gene unpacking from the chromosomes, or in the form of chromatin network, which is unpacked by modification of the histones. Then subsequently we have seen various regulation mechanisms at the stage of transcriptional control, then splicing mechanisms, then translational control, post-translational control, or post-translation modifications, and finally the protein degradation mechanisms. And at all these stages there are various control elements or control mechanisms which is used for regulating the overall gene expression. And so right now we can conclude this topic. Thank you.